بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. نيابة عن رئيسة الجمعية السعودية للكيمياء السريرية دكتورة سامية سكي وأعضاء مجلس الإدارة، نرحب بجميع المشاركين بالندوة الأولى التي تنظمها الجمعية لعام 2021، والتي تحمل أحد أهم المواضيع المهمة للعاملين في المجال المخبري المتعلقة بإدارة الجودة والاعتماد المخبري، وذلك تزامنا مع أسبوع المختبر العالمي. هذه الندوة بالتعاون مع SSCC Saudi Quality Council Healthcare Group. نتمنى لكم ندوة مفيدة وثرية بالمعلومات المهمة لكم للمساهمة بالتطوير في مجال المختبرات الطبية. إن شاء الله ستكون الأسئلة بعد كل سيشن، ونشكر تواجدكم ونشكر جميع المتحدثين، ونشكر أيضا الزملاء بالهيئة السعودية للتخصصات الصحية وكل من ساهم في نجاح مثل هذه النشاطات المتعلقة بالتعليم الطبي المستمر. Dear all, distinguished speakers, moderator, Saudi Society of Clinical Chemistry members and participants, I'm pleased to welcome you all in the first seminar for the year 2021 with the most important topic of all of us as a laboratory personnel and workers, lab quality in collaboration with International Lab Week, which was the last week of April, moved to April 7 due to holy month of Ramadan. This seminar in collaboration with International Federation of Clinical Chemistry and Saudi Quality Council Healthcare Group. Our topic, the first session, and will include standardization, accreditation, and the quality management. Seminars and webinars is an essential component in Saudi Society of Clinical Chemistry efforts to ensure laboratory personnel are updated and well informed. Please put in place all items, elements formulated in this seminar to have a quality lab and share information with your colleagues. At the end of each session, we will receive the questions and answer. And also, just to remind all the participants to register for each session. At the end of our seminar today, we will have a general assembly, and we would like to encourage you all to attend, to listen to your questions and concerns and comments. I wish you all a fruitful seminar. Thanks to all the speakers, Saudi Commission of Health Specialties, and all who contributed in the success of this seminar. Our first speaker, Dr. Graham Pistol. Dr. Graham graduated from University of Glasgow, UK. He is a consultant clinical scientist at NHS Greater Glasgow. He is a former President of International Federation of Clinical Chemistry and Laboratory Medicine, having served as a president from 2009 to 2014. Dr. Graham, he published more than 200 peer-reviewed original articles on biochemical endocrinology and on changing role of laboratory medicine in healthcare. And the first topic will be traceability in laboratory medicine. What is it? Why is it important for patients? We would like to welcome together Dr. Graham. The mic is yours.
Well, thank you very much for that kind introduction, and thank you very much indeed for inviting me to be part of this important seminar. I'm going to talk to you about traceability in laboratory medicine. This is a topic that perhaps is not well understood by people working in clinical laboratories. And yet I hope I'm going to convince you that it is an important part of trying to improve the quality of the results that we deliver to patients. I'm going to start. Oh, now how do I move to the next slide? There we are. Right, I'm going to start with just a few slides of background. This may seem a slightly strange slide to you. We have a clock, we have a pencil, we have the sun, and we have the world. What on earth have they got to do with laboratory medicine? Well, the reality is that no matter where you are in the world, you can tell the time with a very small error associated with it. You can measure length, such as of the pencil, or you can weigh something like the pencil. And again, no matter where you are in the world, you will get results that are very, very similar. And finally, with the sun, you can measure the temperature. And wherever you are in the world, you would get the same result for the same measurement. Now, how can that be? Well, the answer is because all of these parameters, they're measurements that are traceable to international standards. So there's an international standard for weight, for length, for time, and for temperature. And the everyday devices that you use in your home or in your laboratory are all linked to these international standards. So even simple balances for weighing and simple things like rulers and watches and simple thermometers are all calibrated to these international standards. There are more, of course, and listed on this slide here, together with the appropriate international units, Système International, the SI units. And you can see there for mass, length, time, temperature. You can also see amount of substance, where the mole is the SI unit for measuring the amount of substance. And it's perhaps not overstating the case to say that in today's world, SI units underpin our scientific, manufacturing, and technological civilization. Without them, the world would be a very different place. So, what about laboratory medicine? Well, the first problem we face is that nobody really has a clear definition of laboratory medicine. This is just one possible definition of laboratory medicine, breaking it down into a number of subdivisions. And you'll see clinical chemistry is clearly one of them. Now, the problem we have is that patients, and to some extent doctors, assume and expect that all the methods we use will give the same result for a single test, just as if we were measuring length or temperature. Of course, those of us that work in laboratories know that this is not the case and that it's really challenging to make sure that different methods give the same result. Here's an extreme example of getting it wrong. This is from a colleague of mine in the UK, and here it's an EQA sample for anticardiolipin IgG. And you can see there that the individual laboratory results differ by a factor of more than 10, which means, of course, that a mean result across all of those different laboratories has no value whatsoever. And you can really do very little by way of EQA and improving assays when the difference between them is as great as this. Much more commonplace and yet equally challenging is this slide, and this shows the distribution of results in an EQA sample for 7 different methods for measure hemoglobin A2. And the red dotted line is set at 3.5%, and according to the UK NHS Sickle Cell and Thalassemia Screening Programme, that cutoff has been set as the action point in the diagnosis of carriers of beta thalassemia. And you can see very simply that depending on which method you're using, if this was a clinical sample, you would say the patient is a carrier of beta thalassemia or is not a carrier of beta thalassemia. So the clinical decision is determined by the spread of methods. Now, of the hundreds of different methods that we measure in laboratory medicine, I would suggest to you that this is a fairly typical picture, and it is a real challenge for us in terms of translating results into clinical interpretation and usefulness for patients. So I've touched on this already. Why should different methods give the same result? Why do we want the same result from different methods? Well, the primary purpose, of course, is patient safety. As I demonstrated in the last slide, you can mistreat patients if you get the wrong result by using the wrong method. If we get the right result, then patients feel empowered, they will understand their results, and they'll take more care of themselves. And this in turn will lead to greater public confidence in the laboratory system. If all labs get different results on the same sample, then the general public will not have confidence in the work that we do. The second line of 3 boxes is related to quality measurements, if you like, and governance. So laboratory accreditation, the interpretation of clinical guidelines, and clinical governance, if you like, what our employers think of the laboratory, will all be determined by how good our results are. If our laboratory is consistently producing results that are outliers, then we will be regarded in a very poor light by our peers and by our users. And the final box, or 3 boxes, are about informatics. Certainly in my country and in many others, we're now moving to consolidation and networking, where a number of laboratories are networked together. And this can only work if they all have the same reference values and they can get the same results on the same sample. If they don't, then we have a real problem. And then finally, on the right-hand side at the bottom there, we have the electronic patient record. And we're now moving to a situation where no matter where you are in our country, you can have a test done and you can have the result entered into your electronic patient record. Now, quite clearly, if you have a chronic disease that's being monitored, something like thyroid disease, and you get a measurement done in one laboratory, and then you get a result put in from another laboratory using a different method, then there is a real potential for confusion, and you may well find that your management is mismanaged, in fact. So what are the methods that are open to us for reducing between method variability? How can we make methods more similar? Well, there are 2 approaches. The one that you'll be familiar with is on the left-hand side, which is monitoring. And that's the use of proficiency testing, or EQA, to measure how methods perform and to try and move towards consistent performance. And EQA is absolutely essential to the quality of what we do in laboratory medicine. However, it's no matter how good the EQA is, you may never be able to get all methods giving the same result, and that is because they have a fundamentally different design. And the design of the method involves the calibration and traceability to a common reference system. And this is where we're into the area of method standardization and harmonization. And that, of course, is what I'm going to be talking about for the rest of this presentation. So let's move to the second topic, which is traceability in laboratory medicine. Now, I've often been asked to try and explain this in simple terms to people that know nothing about laboratories. And this is the way that I do it. What is traceability? Well, traceability is how we get the right answer. What is the right answer? Well, it's an accurate answer, or the answer that we would get with the best available method. How do I know if the result is traceable? Well, the method has been compared with a better method and adjusted to give the same result. So that's a layman's definition of what traceability in laboratory medicine is. We, of course, are all scientists, so we can deal with it in a rather more scientific manner. And this is the, whoops, whoa, we're going all over the place. There we are. So we can deal with it in a more scientific manner. And what I've got here is the official ISO definition of metrological traceability. And I'm afraid ISO has something of a language of its own, so it's not always the easiest to understand. But metrological traceability is the property of a measurement result which can be related to a reference through a documented unbroken chain of calibrations, each of which contributes to the measurement uncertainty. So for this to work, traceability requires reference materials, preferably certified, and reference measurement procedures or methods in which those reference materials are used. Now, for structurally simple measurements or analytes, it's possible to get pure substance primary reference material, things like glucose, for instance. But for more complex measurands, pure substance may not be available. We can maybe think of a glycoprotein like TSH. And primary reference measurement procedures are usually based on physical methods, for instance, isotope dilution mass spectrometry. Now, if we translate this into something that's a little more easy to understand, it means that we have a hierarchy of reference materials from a primary reference material, which might be pure substance, through a primary calibrator, which should be SI traceable, a secondary calibrator, and then the product calibrator that you use in the method that's used in your laboratory. At the same time, there's a hierarchy of methods or measurement procedures from a primary reference measurement procedure, such as isotope dilution mass spectrometry, through secondary measurement procedures, manufacturers' selected procedures, and then eventually the routine method that you use in your laboratory. And you can put these together in a metrological traceability chain. And if we start at the bottom, you'll see that we're in the routine laboratory where the patient result is obtained using a routine laboratory method. If we move up a little bit, you'll see that the next chunk is in the hands of the IVD method manufacturer. And here they have their own calibrators and their own measurement procedures. And if it's possible to get full metrological traceability, then we move to the top of the chain, where you've got a metrology institute or a reference laboratory dealing with the primary reference material and primary and secondary calibrators and primary reference measurement procedures and secondary reference measurement procedures. And if you have an unbroken chain from the primary reference material and reference measurement procedure all the way through to the patient result, then you have full metrological traceability. Now, the other thing that comes out of this slide is on the left-hand side, if you look at the arrow, as you go up this chain, you increase the level of metrological traceability. And on the right-hand side, as you come down the chain, you increase the amount of measurement uncertainty or error of measurement, if you like. So I hope that's a reasonably easy way to understand what traceability means in scientific terms. And here's another way of looking at it. If we just go to box 5 there, which is the manufacturer selected method, then there are various ways that we can get into that selected method. If you go to the far left, there you've got full metrological traceability through primary reference measurement methods and calibrators and secondary RMPs and so on, before you get to this manufacturer selected method. However, if you're dealing with something that is a heterogeneous substance where there's no pure reference material available, then it might be that the only way you can get to a manufacturer selected method is to have an international conventional RMP or international conventional calibrator, where a panel of international people select a material and they assign a value to it. And then that value is assigned and used around the world. So that's partial metrological traceability. And if you want a simple definition of the difference between method standardization and method harmonization, then method standardization is where you have full metrological traceability. And method harmonization is where that's not achievable, and you do the best you can through conventional calibrators and conventional reference measurement procedures. Well, does this matter? I mean, is this important, or is this just ISO speak? Well, it does matter, because there is a new IVD device regulation in Europe which has to be followed by all the manufacturers around the world. So although it's a European regulation, in fact, it's a global regulation. This was published in 2017 and must be in force in 5 years from then. So by 25th of May 2022, this will be enforceable in law. And this supersedes a previous directive, and it involves changing classification and rules and so on. But the important part of it, as far as traceability is concerned, is in this paragraph 9.3, where it says that where the performance of devices depends on the use of calibrators and/or control materials, the metrological traceability of values assigned to calibrators and/or control materials shall be assured through suitable reference measurement procedures and our suitable reference materials of a higher metrological order. In other words, trying to get as far up the metrological chain as possible, and the manufacturers are obliged to state the level of traceability that is applicable to their particular method. Now, this means, of course, that there are lots of people involved in achieving traceability. You're sitting at the top of this pyramid in a routine laboratory where you've selected methods based on quality performance. But if you look starting at the base of the pyramid, we start with internationally recognized experts who define clinical decision values and analytical requirements. These are then passed to national metrology institutes who will actually provide reference materials and higher order reference methods. These are then listed on global database of reference methods and materials, and this enables standards institutes and accreditation bodies to raise analytical and clinical quality targets. Then we get to a level you're more familiar with, where the IVD method manufacturer produces methods that are traceable to a reference system when it's available. The EQA provider then uses commutable materials to monitor method performance. And finally, we get back to your laboratory using methods that have been selected on the basis of quality performance. So you can see this is a global initiative with many stakeholders involved. I mentioned the word commutable there in the context of EQA providers. Just this slide is, if you like, a slight deviation to explain what commutability means. Here you've got a panel of clinical samples measured by two different methods. And you can see that they correlate very well, and most of the samples lie on a correlation line. Now, if the reference materials or the calibrators that are used lie on the same line, then that reference material is said to be commutable. Sadly, it's quite common for reference materials to be non-commutable. Because of the way that they've been prepared and treated, quite often you'll find that the red blocks, which are the reference material, do not lie on the same panel as the clinical samples. So the reference material is deemed to be non-commutable, and then it's of dubious value. Now let's come on to the next topic, which is the Jctlm, and I suspect none of you know what that means. It's actually the Joint Committee for Traceability in Laboratory Medicine. And it was formed in 2002 to enable a global response to what was then the IVD directive and what is now the IVD regulation. The three founding members were the BIPM, which is the World Body for Weights and Measures, which is an intergovernmental treaty organization for measurement standards. That's based just outside Paris. IFCC was the World Body for Professionals in Laboratory Medicine, and ILAC was the International Non-Governmental Organization for Accreditation Bodies, including clinical laboratory accreditation. Now, Jctlm has grown with 52 members from 20 countries. These include national metrology institutes, EQA providers, professional bodies, and IVD manufacturers. So what does Jctlm do? Well, it maintains a reference measurement system database. So if you remember our pyramid from a few slides back, this is halfway up the pyramid, a database of available reference materials and reference methods. It also coordinates the nomination and review process for new database entries, and it contributes to ISO working groups on reference systems. It holds a biennial scientific meeting, and the next one is scheduled for December this year in Paris. And it also provides educational materials to support an understanding of the importance of traceability in laboratory medicine. This is what the database looks like if you follow that URL, and you can type in an analyte name, and then you can have a look to see what reference materials or reference methods or reference measurement services, that means an international reference laboratory network, to see what's available for that particular analyte. And at the moment, Jctlm database has 302 reference materials, 188 reference methods, and 161 reference measurement procedures. Now, that sounds fairly impressive, but in fact, there are, at an estimate, there's somewhere between 3,000 and 4,000 different things that are measured in clinical laboratory medicine. So we're probably only about 10 to 15% where we have certified reference materials or reference methods. Some of you may be familiar with the World Health Organization. That has a catalogue of international reference preparations. There is no traceability status attached to this catalogue, but many of these reference preparations from WHO are the basis of some of the assays that we use every day in our laboratories. So they're valuable, they're conventional calibrators, but the traceability status of them is largely unknown. Now, where are we therefore on standardization and harmonization of methods? Well, here's a very simple summary. We've got the number of methods up on the y-axis and the complexity and time of money in achieving method standardization and harmonization on the x-axis. And you can see that we've already made good progress with small molecules such as glucose and simple peptides. But once you get into the area where you're into complex proteins, or even worse, when you get to areas where there's biological variability, such as nucleic acids or cells and microorganisms, then the amount of time and effort that's involved to achieve standardization and harmonization of methods is really quite formidable. And it's a really massive challenge for us to achieve this across the full range of methods that we use in laboratory medicine. If you want to get more information on Jctlm and traceability, this website provides you with lots of educational support material, and you can find webinars and scientific presentations like this one that will help you with your understanding. So I've just got a few slides left now, and firstly I want to share with you a case study. And the case study is of an analyte that will be very familiar to you, and that's hemoglobin A1C, which you know has been established for more than 20 years from major clinical trials as the key analyte for long-term monitoring of diabetes. Now, IFCC published a reference method based on mass spectrometry of a signal hexapeptide way back in 2004. And following that, a global IFCC reference laboratory network was established to help people ensure that methods are all aligned to the IFCC reference method. And international consensus, including from all the diabetic organizations around the world, recommends only using IFCC aligned methods. Why is HbA1c so important? Well, I think you'll be familiar with this, that if we can reduce HbA1c by 1%, we have a dramatic impact on reducing deaths, microvascular complications, and myocardial infarction amongst diabetic patients. Now, there's a huge literature on HbA1c, which I've summarized in this single slide. Before method standardization, the between method variability of HbA1c methods was well over 20%. Following method standardization in 2004 and the recommendation that everyone should use IFCC aligned methods, between method CV for HbA1c method has now fallen to 5% or less. Now, this improvement in methods is so good that the World Health Organization has now said that you can use HbA1c to diagnose type 2 diabetes, as well as to monitor the response to therapy in diabetes. And the global network that's been established is driving up quality improvement in HbA1c methods, and has recently published global quality metrics for HbA1c assays. And I'll share just one example in this slide. This is from routine laboratories in multi-center study across China. And there you've got different results for samples where the HbA1c concentration is denoted by the color. What you need to take from this slide is firstly that all of the methods and all of the results are bunched very closely together, and that there is virtually no bias, and that the between method coefficient of variation is around about 3%. Now, if you translate this into sigma metrics, it means that virtually every laboratory is doing at least better than 2 sigma in terms of sigma metrics, and many are now at the level of 3 sigma metrics. And that's really extremely good performance across routine laboratories and a range of different methods. So that shows what's achievable when you get full metrological traceability for a very important clinical biomarker. So finally, what can you do in your laboratory? Is traceability just something that affects other people, or does it also affect you and the results that your laboratory produces? Well, in very simple terms on one slide, I'm suggesting this is how you can help to promote traceability. Firstly, you can check the traceability status of the assays your lab uses. This means looking at the very small print in the instructions for use of your methods. Remember, the manufacturers are actually obliged to give you the traceability status of the methods, but they usually do it in extremely small print. And you may well find that they are not as traceable, the methods you use, as some other manufacturers that offer similar methods. If you want to know more about traceability in laboratory medicine, you can go to www.jctlm.org and you'll find resources to help your understanding. Equally, if there are important biomarkers where you know from your EQA schemes that the between method variability is too big for an important clinical biomarker, then you can make suggestions for how we can improve that by producing clinical reference materials or clinical reference measurement procedures. And there's now an organization that coordinates these suggestions and puts together panels to actually start making the reference materials and the reference methods. You might want to incorporate a whole session on traceability into the training of laboratory medicine specialists in your country. Usually there's very little in there in normal training. And then finally, you might want to have more than just me speaking. You might want to have a more detailed consideration of traceability in a national conference, in which case JCTLM will be very happy to provide you with a range of speakers who can cover a range of different topics. So that's been a very quick race through the whole idea of traceability in laboratory medicine. And I hope I've convinced you that not only is it important for you, that the fact that we are trying to get results from different methods to give very similar results is also extremely important for patients. So thank you very much, and I look forward to questions a little later on. Thank you.
Thank you, Dr. Graham, for your excellent presentation and very important, actually, topic for us as laboratory personnel related to traceability in laboratory medicine. And now we can move to our next topic, and we will leave a question at the end of our session. So our next speaker is Dr. Anwar Burai. Dr. Anwar, he is a section clinical chemist at King Abdulaziz Medical City, Jeddah. Dr. Burai, he is the Director of Clinical Laboratory Sciences, King Saud bin Abdulaziz University for Health Sciences and Jeddah. And he is also a member of Laboratory Council and Scientific Committee of Saudi Commission for Health Specialties. Dr. Anwar, he is the representative of our society, Saudi Society of Clinical Chemistry, with the International Federation for Clinical Chemistry. Dr. Anwar will discuss the laboratory calculated test, validation, and implementation. Anwar, and the mic is yours.
Thank you so much, Dr. Ali. Thanks for your introduction. Thanks for the Saudi Society for Clinical Chemistry for their invitation. Today I'm going to talk about a new subject, which, frankly speaking, not a lot if you are going to do or search for such topics, you will find but a little literatures are available for. The topics called laboratory calculated test and validation and implementation. We talk about a brief introduction, then we'll talk about the types of calculated test. Then I'll talk about the quality control methods or procedures we follow for calculated test and the available validity testing if you want to do. Also, I will mention the CLSI and CAP, how CAP reports usually done on calculated test. Then we'll go through the implementation and then we conclude our presentation. As we know that
Dr. Anwar, Dr. Anwar, sorry for interruption. Actually, the voice or the sound is a little bit interrupted and weak from your side. If you can double check.
Okay, yeah. I'll try my best to increase my voice. As we know that calculated test is very important in different laboratory sections and in laboratory. You can find it in clinical chemistry, you can find it in hematology and serology, in all different sections of clinical laboratory science. But unfortunately, the methods for validity, the QCs applied to calculated tests are not that well presented or well available. In general, the patient results can be obtained by either direct determination of requested test by various clinical methods, as we know, or it can be calculated using unknown test results based on relationship between different measured tests. The calculated laboratory test can be defined as the outcome of numeric calculated results, which usually provided by
Dr.
the
Anwar.
in order to.
Dr. Anwar, sorry again for interruption. Can you, I think something wrong with the mic, your mic. You can connect and disconnect the mic. The voice is still in, there is interruption in your voice. There is no voice from your side, Dr. Anwar. Dr. Anwar, are you still with us? Dr. Anwar? There is no voice from your side, totally silent, if you can hear us. Dr. Anwar, there is no voice, totally silent from your side. Dr. Anwar, totally silent from your side. If you can double check, please. Totally silent. Our colleagues in control room, can you check please the status of Dr. Anwar? Still with us or disconnected? We are still waiting for Dr. Anwar to reconnect. Mr. Ali Al-Hamid, if you are with us, please can you check the status and give us the feedback? I think just we have technical problem that we will give 2 minutes for Dr. Anwar to join us back.
نسمع الصوت؟ سامعين الصوت؟
Dr. Anwar is back.
سامع صوتي؟
Yes, very clear, Dr.
طيب اوكي. Can you put the presentation please again?
Okay.
Okay. Sorry for the interruptions. We'll go and continue our presentation. So the calculated laboratory test can be defined as the outcome of numeric calculated tests provided by the laboratory in order to interpret, so a medical decision can be taken about it. The calculated test is based usually on different number of numeric variables. These variables can be called the components. These components are actually incorporated into a scientific equation. The number of these incorporated tests or components can be either 2 or more tests. So the calculated test can be calculated. This is just an example of clinical laboratory test. We have a lot of in chemistry, for example. We can do the adjusted calcium. We can do an ion gap. We can calculate the osmolar gap and osmolality. So all of these are examples of different examples in chemistry or in clinical chemistry calculated test. As you can see, it can be the component can be either 2 components, 3 components, or sometimes 5 or 4 components. Renal clearance, for example, eGFR, the creatinine clearance. The eGFR, as you know, we have different equations. Cockcroft equation, modified MDRD, CKD, ABI equations, all these different equations that can be utilized in clinical laboratory. Lipids, for example, Friedwald equation, as we know, is very common equation that we utilize to calculate the LDL test, as you know. Also in hematology, we have a lot of indices can be calculated. These calculations are very important to take a medical decision. The calculated test, as we say, has a definitive clinical outcomes where there is no single measure test can be done. This is just only one of the types of the calculated test, where the decisions the clinical decisions will be taken according to the outcome of the calculated test. For example, creatinine clearance. When there is a decrease in creatinine clearance, that means there is a renal failure. When there is an increase in INR, for example, in hematology or coagulation, then there is a business of warfarin, for example. So the blood clotting prevention will be lasted for a longer time. Decrease in MCV and MCHC that indicate a microcytic hypochromic anemia. This is one of the outcomes of one of the type of calculated tests. So the first type of calculated test, where there is a clinical outcomes, there is a solid clinical or definitive clinical outcomes, depend on these type of clinical or calculated test. Another type of calculated test, where there are calculated tests which has almost the same meaning or the same diagnostic value of the measured test. We utilize mostly this type of calculated test as cost-effective test. For example, LDL, instead of measuring LDL, we can calculate the LDL. Instead of measuring ionized calcium, we can measure the adjusted calcium by using a simple equation. Instead of measuring free testosterone, we can measure the free androgen index. And instead of measuring osmolarity, we can calculate the osmolarity using the sodium results. So all of these calculated tests are simple, can be applied simply in the lab without any cost or extra cost. Another type of calculated test, where the calculated test can enhance the diagnostic value of the measured test. For example, measuring PSA or free PSA alone, it doesn't mean a lot when compared to the free PSA ratio. When there is a urine protein, for example, and urine creatinine alone, it cannot give you a meaning without measuring the ratio between protein creatinine ratio in urine. For aldosterone and renin alone. So if you want to measure each individual test alone, it doesn't mean a lot when you measure the calculated ratio of these components or of these measured tests. Creatinine, for example, alone is different from the measurement of the eGFR. Free kappa or free lambda also is example. So when you measure kappa lambda ratios, give you enhancement in the diagnostic value, for example, in multiple myeloma. For test validation, if you want, as you know, Graham in the previous lecture, he talked about one important issues for tests to be standardized. Also, the validation is important. Is a part of the standardization is a component of validation. For example, for validation, we need specificity, sensitivity, accuracy, precision, linearity, and reference intervals. All these components are important for any test to be validated. For laboratory tests, the problem with it that the QC program and the regulation deal with these calculated tests only applied or available for measured tests. For example, if you want to go and see the regulations about the measured test, you can apply the QC program using Westgard rules, Sigma. You can find the regulation in CLSI and CLIA, but unfortunately, there is no solid guidelines for the calculated test. Calculated methods are usually another issue, that the calculated methods are usually clinically validated, but not analytically validated. This is one of the problems of calculated tests. Another problem with the calculated test, that the calculated methods are not standardized, as Dr. Graham mentioned in the previous presentation, that some measured analyze, or many of the measured analyze, not standardized. So what about calculated tests? Calculated tests are utilized for clinical decisions. It can be used, the outcomes can be used to take a decision about the patient, exactly the same as measured tests. So standardization also is one of the problems, is exactly the same as measured tests. For example, if we look at the adjusted total calcium, or in presence of low albumin, you can see many equations. Since 1971 up to 2000, there is about 4 equations. Which equation is suitable to me? Which can I apply in clinical laboratory or in my lab? This is one of the problems of the calculated test. If you go through the CLSI standards, you will find a lot of guidelines, a lot of rules and regulations, but the problem is still there is no solid guidelines regarding the calculated test. When I looked here and there about calculated test and quality control, I found some published papers talking about the quality control of calculated test, published in 2005 and 2017, is scattered here and there. But they are just only small, published in small quantity actually. But at least it gives you an idea how important to establish regulations for quality control for calculated tests. So when I look at these regulations, they say that the calculated tests need to be dealt with exactly the same as measured tests. It needs a leveraging chart. It needs a QC to be established, the mean. It needs a standard deviation and coefficient of variation to be established. We need to deal with it exactly the same as measured tests. So to do this, we need to look at the components of each equation and look at these components and measure it for 20 days and estimate the mean and the standard deviation, so that you can obtain the 2 standard deviation or 3 standard deviation to construct the leveraging chart and apply plotting exactly the same as you can see here in this table for 20 days until you get this SD and the CV and the mean to establish your leveraging chart. As you can see, for example, this is just an example to show you that even the components, even if the components of the equation are within the 2 standard deviation plus or minus, you might get outliers from the calculated test. So this way, in case you get outlier, then you should not utilize the calculated test in that particular day. So we have to be careful about this. We have to establish our own QC program for calculated test, exactly the same as the measure test. The CV, there is some regulation. There is one advice that whenever you want to make a precision study, you make sure that the CV of any measured components must be lower, so as to obtain an acceptable CV for the calculated test. The CV must be lower than must be lower than the CV of each incorporated test. So this is one of the regulations that we have to be careful about, because if the CV of the calculated test is less than the CV of each incorporated test, then there is a chance a big chance of unreliability in the application of the calculated test. Regarding the linearity, there is no strict regulations about or guidelines to tell you how to do the linearity for the calculated test. But at least we have to be careful about the linearity for the calculated test and check the minimum and the maximum value for each component of the equation, which can be reported in the equation when established. Otherwise, the equation should not be utilized and the equation should not be valid. For example, for LDL calculations, we have to be careful about triglyceride not to be more than 400 or 4.5 millimole per liter. Otherwise, the equation will be invalid. For the analytical reportable range of each component, they must be considered. Because, for example, if you want to calculate the anion gap, and you can see that sodium or chloride or bicarbonate exceed the reportable range, then the equation should not be calculated and the result should not be reported. The same story with the reference interval. As we know that the reference interval are very important, they have to be verified. You can check the same regulation for the measure test, how these regulations applied. If you want to verify the reference range, then for a measure test, then you need to verify the reference range for the calculated test. The same regulations use 20 samples. The components of each equation should be within the reference range. Then the calculated test will be verified. Or you can make estimation for the reference range, or establishing a reference range. That means you need to have more than or equal to 100 individuals to calculate the each component reference range, and then you can estimate the reference range for the calculated test. For example, for adjusted calcium, if you want to estimate the or validate the reference range, then the 20 sample for total calcium and albumin should be within the reference range to verify the adjusted calcium. If any of the components difference or values outside the acceptable range, then they should not be included in the estimation. So the equation cannot be verified for reference range. Also, one of the important things, which is the clinical validation of calculated test. This is the if there is no chance to do analytical validation, at least you can, or you have to do the clinical validation for calculated test. Clinical validation is the determining the ability of a test to diagnose or predict risk for particular health condition is using clinical laboratory in case there is no comparative method. They are common, or the clinical validation commonly used for calculated tests. Usually, for example, if you want to use EGFR equation, then you need to get, for example, 10 healthy subjects. If the 10 healthy subjects their EGFR is more than 90, then you can validate the equation for healthy individuals. Also, maybe you can use 10 abnormal or 10 samples from patients with renal failure. If the result is very low for EGFR, then you can, by this way, validate the EGFR equation. For CAP proficiency testing, recently the CAP stopped in 2020, in October, they stopped releasing the calculated test for LDL and other calculated tests. They put a comment that for calculated LDL cholesterol is not possible to construct appropriate peer groups because of all possible instrument reagent combination for the 3 levels components used in the field equation. So because of the combination different from different companies, they cannot make a validation for big groups, and that's why they advise all participants to, as they said here, you should verify that your calculation was done correctly according to the equation, so they are not anymore include calculated LDL in their proficiency testing or any other calculated test. And the reason, as I said, because of the variability in the number of instruments and number of reagents involved in the equation. This is another regulation they say calculated values reported with patient results are reviewed every 2 years or when system change is made that may affect the calculation. I think every 2 years is a long period of time. I think the calculated tests need to be validated exactly the same as measured tests, especially with the increase or in the recent advances in the automations and the recent advantage, the advantage of the softwares. We can incorporate the calculated test together with the measured test. So I think 2 years is too long period. We should work hard to deal with the calculated test exactly the same as the numeric test or as measured test. For calculated test to be implemented, usually the lab receive order to implement calculated test. Whenever there is an order received, the lab should liaise with the related department. For example, if you want to implement in your lab EGFR equation, you should negotiate, or you should sit down with the nephrology department and see what kind of equation is most suitable to them, and then you select the appropriate equation to be implemented in the system, and then this equation should be constructed in the system, and you use or select to implement the equation using either the laboratory information system software or implement the equation in the middleware. So it depends on each site that you decide to. Each one has a limitation or advantage. So you need to look at your advantage and limitations, where to construct your calculation or where you construct your equation, either in LIS or in the middleware. This is can be left to the lab who to decide which one to select. Then after selecting the site of implementing the equation, then the method and technical validation should be started, where the equation applicability, the reference range validation, the cutoff value of validation, the quality control limits, and also define the sources of error which might cause an interruptions or cause the disturbance in the releasing the result of the calculated test. Also the precision, linearity limits, and clinical validations all are important to be looked at, and make a validation as much as you can, because this is, as I said, this is a medical decision. The outcomes can be used to treat the patient exactly the same as a numeric test. In summary, we can say that when calculated test results become reliable, they become more convenient and cost-effective to calculate such results than to measure samples using reagents and equipment. The current method, validation and quality control regulations are not enough, but we need to pay attention to the clinical outcomes and decisions of our equation. The guidelines must be established. There must be an established guidelines for calculated tests to regulate their utilization as any other measure the test. Thank you so much, and hope to receive your questions by the end of this session. This is some references, and thank you so much for your listening and for sorry for the interruptions regarding the sound at the beginning of this presentation.
Thank you, Dr. Anwar, for your excellent and informative talk, and we would like to apologize for the technical issues that we faced during the presentation. And again, I would like to remind everybody that to write down their question in our chat in order to be presented for the speaker at the end of the session, inshallah. Our last speaker for the first session is Dr. Eng Peter Diemen. Dr. Diemen, he obtained PhD. He is a doctor in medical science degree at Medical Department of University of Antwerp in 2003 and executive MBA from the Antwerp Management School in 2013. Dr. Eng, he joined Biorad Laboratory in 2014 and currently holding a position of International Scientific and Professional Affairs Manager at Biorad Quality Control Division, Irvine, United States. He works with the clinical laboratories and societies around the world in developing, educating, and promoting quality control concepts, strategies, and best practices. He is a member of European Federation of Laboratory Medicine Working Group Reanalytical as expert consultant, and he is also a former member of Total Error and EFLM Task Working Group. Dr. Diemen will present a topic perspective on quality control in laboratory 2021 and beyond. We would like to welcome together Dr. Diemen, and the mic is yours.
Thank you, Mr. Chairman. I just wanted to check if the sound is okay.
The sound is excellent.
Okay, thank you very much. So I would like to thank Mr. Chairman for the kind invitation to speak to you today. Also, thanks to the Society for this nice opportunity. So my name is Peter Diemen, and I'm speaking to you from Belgium, Europe. I'm living close to Brussels. For some of you who know Belgium, I'm living in a place between Brussels and Antwerp. And actually today we are in the month of April. We still had snow today here in Belgium, but that's just as an introductory topic. So I'm very happy to speak to you today on quality control. And I would like to give you a kind of perspective on quality control on the eyes from a quality control perspective on the design of quality control products, and also what are the topics for the future if we are looking to the production and design of quality control products. And that's the reason why in my title I also have 2021 and beyond. So at the end of my presentation, I also hope to give you some perspectives on the future topics that will be and are already important today. So in the objectives, like I mentioned, I would like to share with you the purpose of quality control. It's a topic we all know what is the reason behind, but I would like during this presentation discuss that with you almost from a philosophical perspective and show it to you with the eyes how we are looking to the production and design of quality control. I also would like to highlight to you the different types of quality control, and at the end, as I mentioned, some updated trends and perspectives 2021 and beyond. But let's start with the beginning. Why are quality control materials used? And I think a lot of you will know the intended use of quality control products is to verify the reliability and medical usefulness. So that's, I think, a very simple and solid approach. Another aspect is if we are designing and generating QC data, we also using statistical process control to interpret this data and also to have a kind of process approach. Because as you all know, we cannot put one quality control next to each patient sample. So we need to find a way of optimizing our QC approach, and therefore we need statistical process control. Reliability, just to start with the definition, as we all know, reliability means exactly the extent to which an experiment, a test, or a measuring procedure yields the same results on repeated trials. Medical usefulness, of course, that's important that the test results are appropriate for diagnosis, prognosis, and treatment. And as I will show you also in my following slides, if we are designing and using quality control products, it's also important that we are having this QC products around the medical decision limits. Like I already mentioned, it's important to use these quality control alongside with the patient sample. That's very basic and very simple, but also very important. And I think also something to take into consideration is the aspect that we also want a quality control taken through all the steps of the analysis. And as some of you know, a decade ago, 2 decades ago, we were focusing a lot on the analytical step, so the analyzer, the analysis itself. But now, during the last decade, and still nowadays, there are a lot of publications and studies showing that, of course, the pre-analytical and also the post-analytical steps are also very important to take into consideration. And certainly also, I will show you that later on, during the pre-analytical phase, it's important to also have a good quality control approach. But also during, for instance, extraction phases, molecular testing and things like that, it's important that we are trying to have quality control during all the steps of the analysis. If we are now looking to the ideal quality control, of course, we are not living in an ideal world, but let's think about what's the ideal quality control material. It's closely mimicking patient samples. That's important. Has medical useful measurement concentrations. I think that's what we just discussed, very important. And can undergo the complete analytical process. Now, if we are looking to what the material, the QC material is, and this is a very high-level overview, but I think most of you will know that the main components of a quality control material are 3 points. It's first of all the matrix, as we call it. So that's the solution containing the measurement. Then we have the measurement itself. I think a very important, of course, aspect is the measurement itself, like chemicals, enzymes, proteins, as far, we want to measure and also control specific analytes. And then preservatives and stabilizers, if needed. And I can imagine that you think, okay, preservatives, stabilizers, it sounds a little bit chemical. Maybe some of you would interpret it as something negative. But at the end, what we want to do is design a product that also gives you the stability over time and also give you the usefulness that you not every day need to use another product, so that you also can minimize the different types of variables during your process. And that's the reason why we are also, if products are designed, need to be able to stabilize the product and also have preservatives that, for instance, the shelf life or the open vial stability of your product has enough length that you don't need to do every week or every few days a crossover study, because crossover studies or studies where you need to go from one product to the other also bring variability in the process. So important to know about these 3 components, but certainly also why these are important. The matrix, it's of course not only the movie as the matrix, but here the matrix in essence can be serum, plasma, urine, like you read, whole blood, body fluid, spinal fluid. Of course, there are also other non-human matrices. Of course, from our perspective, we try to have the human origin matrices or as close as possible, but there are also other matrices like chemically, bovine, aqueous. So when you are choosing a quality control product, and we come to that later on, it's also important to really have a look, okay, what is now the matrix of that specific quality control product? Concentrations near medical decision limits. I think that's a very basic and very logical topic to discuss. I do not think I need to go too much in detail. I think we are all aware, and it seems very logic, that of course we want to have our quality control product around the normal physiological values. Of course, we can also have the abnormally high or abnormally low concentrations, but it's important certainly to capture and to control the normal physiological values where our quality control, where also our measurement is taken place. Of course, we will see later on in specific curves, it's also important not only to have one point in the curve, but also have a broader range. But we come to that later on. Now, one question for you in the laboratory and for a lot of us is how we make a choice of the different types of quality control materials. And actually, to be open and honest, I do not think it's an easy thing to make a choice. That's one. Two, there is also not one golden rule or one size fits all solution. So in a way, it's something you or your colleagues in the laboratory will need to take attention for specific aspects in making the choice for product A or product B. And with you, I would like to share with you some perspectives. What could be these factors we are using in the choice we are making? From a very high level perspective, I think the source, composition, expiration date, and stability, it's a very important one, but a very pragmatic and basic one, and other features. And I will go in the following slides through some possible criteria that can help to make a choice for a specific quality control. First of all, one thing we hear a lot about, and also is a very important aspect, is the first party, second party, or third party quality control products. And these are definitions and terms that are used quite a lot. And you will see even in ISO 15189 and in other more recommendations, the aspect of a third party or second party or first party quality control is also described. From a more general perspective, it's also good that you choose a quality control product that has no potential bias for a specific platform or analyte. And I will explain myself a little bit more in detail in the following slide. So of course, if you have a quality control product that is provided by a manufacturer, it's important to know that it's designed in an independent way from, for instance, the calibrator of your product or from certain reagents. So here in this very basic and simple graphical representation, it's important if a quality control product detects something that it also detects every time a certain shift or a certain error. If there is, of course, due to the composition of the quality control product, a certain potential bias, then the quality control will not do what it needs to do. So there it's important also to see what is the source of the quality control product, and also is it designed and developed in an independent way. So here an overview. I will not go into all the details, but these are more the basic definitions of a third-party quality control and a first-party quality control, where a third-party control is manufactured independent of the instrument manufacturer, and in that way we'll have a better estimation on how the system is performing. So in that way, you are sure there is no potential bias in the quality control itself, and also in what the quality control product will give you as a result. As I mentioned before, the ISO 15189, we all know the ISO 15189 is now under also revision, but in the current ISO 15189 in clause 5.6.2.2, Note 2, there it's clearly mentioned use of independent third-party control materials should be considered either instead or in addition to any control materials supplied by reagent or instrument manufacturers. So I think also in other guidelines like CAP and also local guidelines, these aspects are certainly highlighted. And I also think from that perspective, it's important to take this into consideration. Now, like we mentioned before, if we need to make a selection, it can be influenced by the matrix material. So the human matrix, and I think that's a very logical statement, is preferred. Measurement composition should include the measurements of interest, of course, and also should be at important medical decision limits. That's a topic we just discussed in the previous slides. I think long shelf life, it's not only something just to make it easier for you or for your buying cycle, but I think also the open vial stability is an important factor, that the analyte is stable, and in that way we can have over time also a more stable process. Like I mentioned to you before, if we have a longer shelf life and greater stability, in that sense, we don't need to go from one quality control lot to the other. And that's the same with if you change from reagent lots or things like that. It's important to have as much stability in your process as possible. And of course, if you need to go with crossover studies or others, these are not only time investments, also not extra costs, but in that way, the longer you can keep your process stable, also from a quality control perspective, that helps you also to bring more stability into your control. So like I write it here, new QC materials will also require expensive crossover studies to establish or verify the target and ranges. So in that way, the more stable products you also have over the long run, they can give you also a good management of your processes. How many QC levels? Of course, that's also a very important topic. We just also in previous slides discussed that. I know these are very basic charts. I cannot go in all the details and all the nuances, but I think we all feel very pragmatically that 2 levels are good to check a linear calibration curve. Of course, multi-point calibration often need 3 controls to cover the full range of measurement. This is, as such, not a very simple topic. I know there are more nuances and details behind this, but I also thought it's good to at least share with you, if we are giving the overview of the choices of a quality control product, that the aspect of the levels is an important topic to take into consideration. Another important aspect, and now I'm switching a little bit the gears from, we have the product itself, the quality control product, but also the statistical process control is important. It's not only the value, but also the way how we interpret the values. And I think the interlaboratory comparison programs also not only track the performance, but can also give a comparison to your peers. And I think for all of you who are already using this interlaboratory programs, I'm sure it helps you to also manage your QC data. And the QC management data is also important, that because it can give you access to specialized or more specific features, basic features like also the previous speaker was showing Levy-Jennings charts and other more statistical visualization and representation. That's one. And also data storage. It's good to have a good system that not only can capture the data, but have a good way of traceability and also tracking the data over time. When you have an audit or when you have need to check earlier data, it's also good, like with a lot of systems we nowadays have also in other, let's say, type of businesses. Also for QC data, it's important to have a good QC management software. So we also think in making the choice in a specific product, it's also good what type of data management programs can help you. and in that way can also make your life easier in the lab. It's important also to have a view on the aspect on which platforms do the quality control products need to be used. If you only have one type of platform, okay, then it can be easy also to choose a specific quality control material. But also be aware that independent QC materials, like I mentioned here on this slide, can be used across many instrument platforms. And that's a topic, as nowadays we are sometimes not only working in our own laboratory, but we are part of a broader group of laboratories, or even in our own laboratory, we have different platforms. That could also be a reason to choose a specific quality control product that can be used on the different platforms. And of course, if you have a quality control product that's designed for a specific platform and cannot be used on another platform, that could give some challenges. So in that way, that's a topic, and I will discuss that also at the end of my presentation, where we are also doing internal and external research, because the quality control approach, not only the products, but also the data management approach, is good described, and I think also in the literature, also enough research has been done. But one of the interesting topics to further explore is to, if you're working with multiple platforms or even in groups with platforms at different sites, is how do we manage not only our operations, but also how do we manage our QC design? And that's not the topic of today. I could give a lecture on its own on this topic, on the quality control for multiple platforms, but that's certainly an interesting topic for the future, where you see that more and more platforms are used across different sites, in groups or in one big group. We still have different types of platforms. That's something to operationally manage, and also in a pragmatic way, not only the theoretical approach, but how do we do it in the real life? That's a topic that also is interesting to further explore. And certainly in the future, a lot of new insights will become clear. So just coming back to selecting quality control materials, it seems a very basic and simple question, but as I try to explain and share with you, there could be some specific variables you can and will need to take into consideration. Of course, we know you cannot take into consideration all these aspects, but it's good, if you need to make a choice, certainly to look at it from different perspectives. And in this way, I made an overview of what we just discussed, the first and second third party, human matrix, stability, interlaboratory comparison programs. So I will not read them all, but I think all these aspects could be good to take into consideration, or if you need to make a new assessment, to evaluate what the pros and cons of specific materials are. And I'm certainly aware we are not living in the ideal world where you will have the ideal quality control product and quality control approach. In real life, sometimes it's making, like, if we are designing new products, we also need to make sometimes certain trade-offs, sometimes compromises. Compromises, I don't want to, it's not a negative thing. Sometimes we need to balance and see what is the best pragmatic approach. And I think also for you in the laboratory, when choosing a specific laboratory quality control product, these factors can play. And I think all of them, or most of them, you already knew, but I tried to give you, at least with this lecture, a kind of overview. Now, just to the last chapter of my presentation, I wanted to share with you some topics I think that are already today very important related to quality control and quality control products. But I'm also sure that in the future, if we will go to meetings, and unfortunately now we only can do virtual meetings, but I hope in the future we will certainly have again face-to-face meetings. And these are topics that certainly will come back in keynote lectures or other research, or certainly other discussions around quality control. And I would like to go through with it. It's my perspective on topics that I think are very important and certainly will become even more important for the future. The first one I would like to share with you is patient risk management. Here you see my overview. I had a little animation in it, so you see them now all together. But let's focus on the first one, the patient risk management. I think the work that we are doing in the laboratories, the patient is anyway central. We are doing it for this patient because finally physicians, clinicians will need to make a diagnostic decision or a treatment decision based on the data we provide. That's important, and that's something we all are aware of. Of course, certain analytes are more or less critical for a patient or imply a certain risk if there is an erroneous result. If there are erroneous results of specific analytes, one analyte will be more impactful for the patient than another analyte. So there, the aspect of the patient risk management is an important factor. And risk management is already something we are doing for years. But I think nowadays, like I mentioned to you before, where in the past we were looking to our analyzer, the analytical process, I think now bringing the patient into the equation more, I think that's an important topic. It's already important, but will be more and more important. If I'm now making the link to quality control, I think there we will also see that we even can fine-tune for each individual analyte, potentially the quality control approach. What do I mean with that? Is that for certain analytes, maybe we are nowadays over-controlling, but for others, maybe we are under-controlling and could be optimizing this. So it's not just that we need to do more or less control, but maybe for specific analytes we can do more and for others less. And that's all based on what is the potential impact for the patient if there would be an erroneous result. So in summary, I think this topic of fine-tuning the quality control approach for specific analytes, and we all know that, for instance, troponin or LDL or another analyte can have different impacts and consequences for the patient. If we are in an ER or just in a maintenance of a certain patient, that's all the difference. So I think that their data management software with specific tools can help us to do that, because we cannot for each individual analyte spend 10 minutes to optimize. But if we have the tools, also the platforms to help us with it, I think that's the next step for the future, where we can approach this in a more efficient way. A second topic that you see on my slide is the cost of quality, because cost is something we all need to live with. We all have budgets we need to make, and we need to stay within our budgets. And if you are talking, or we are talking to CFOs, sometimes they see, as we all know, quality control or quality as sometimes more the cost than what it could give us. And I think preventive and maintenance costs are certainly important topics that we need to take into consideration, in that sense that the cost we are investing for prevention and maintenance can sometimes be much more or less better than the final failure costs. Because if we have failure cost lawsuits, and unfortunately even in the last few weeks, you see in the news publications on labs where something really went wrong, or really there was a big impact to the patient, and that's something we don't want. So there it's important also to have a good balance, but it's also in, let's say, to try to convince that we also need a specific budget for the quality control. And the CLSI recently also published their updated guidance on cost of quality, where they also gave some specific tools, some, let's say, sheets, calculation sheets, to give you a pragmatic, easy way of doing some basic calculations, and in this way also could give you some insight in the specifics, because it's not just a theoretical thing, cost of quality, but with some basic calculations for your individual lab, you can also have good and specific insights on where you could build the knowledge, that's one, and also build some more understanding on what you could do more specifically, and in what case you could also support the investments needed. Again, this is a topic on its own. We don't have the time today to go into the details, but I think that's an important topic we see more and more also discussed in the near future. Pre-analytics, that's another topic that I think is important. Pre-analytics, post-analytics. A lot has been published on this already over the last decade. We are all convinced it's not only our analytic center point, but also the processes before and after are important. I give you here an example of, for instance, QC of serum indices. This is a topic we are also working on at this moment, but there are also other approaches where I think the pre-analytical steps, also from a QC approach, needs and will get more and more attention. Molecular tests. Molecular tests I just gave here as an example. There are also other tests. It's not only our chemistry platform anymore. We have specific technologies, different technologies in our lab that all need a specific quality control approach. And also with molecular tests, for instance, we have the classical in-kit controls, but in-kit controls in most of the cases are not the same than third-party controls. For instance, in-kit controls cannot always give you all the steps, the extraction steps and things like that. So there it's important with new tests, like with molecular tests or other test technologies, also to see always what do we want to control, what are the different phases we want to control, and to have a kind of holistic approach to it. And certainly with molecular testing that gets more and more popular, more and more also developed, I think a good quality control approach is also important there. For instance, following the CQ value in a Levy-Jennings chart, that's something we see more and more, have specific goals for the CQ, the CT value. So these are things we are also and will be seeing more in the coming years and even decades, I think. My last point to conclude for today, I think, is efficiency and ease of use. And that's something from our perspective that's very important, because we also not only thinking what is the best optimal quality approach for a laboratory, but we also try to see how can we make your life, to say it very simple, easier and also more efficient. Because if you have too many, for instance, manual steps in the process, that's something that takes not only extra time, but also can bring extra variability or even errors. So that's, for instance, controls in a tube, to say it very simple, is if we can make the individual intermediate steps or take some of these steps away, that, for instance, you can load tubes in your analyzer and you don't even need to reconstitute, or you only just load the tubes, and in the tubes the quality control is already inside, and it can, with some platforms, stay inside your platform, so you don't need to, everything can be controlled with the software, and you don't need to take all these manual steps. These are things that not only making your life easier, but like I also wrote here in the slide, less time, less errors. So this is optimizing the workflow. I think that's a topic. It's also not something new. The workflow analysis is something we know already for decades, but I think also in our field of laboratory management and workflow management, I think this is an important topic. We will see more and more, let's say, new developments, and we think it's important to have these more optimized for the future. So with this, I know these last considerations, I went through them or observations and trends quite quickly. For some of these, we can have 2 or 3 extra lectures. Of course, we will not do that today, but at least from my perspective, I think these topics certainly in the coming months and years will be also topics that are related to quality control and will get more and more attention. So with this, I would like to thank you all, even though it's virtual today, would like to thank you for your attention, and maybe I hope in the future that we could have maybe a face-to-face meeting in Riyadh or Jeddah or at another place, but I would like to thank you for your attention. And I think I give the floor again to the moderator for potential questions. Thank you very much.
Thank you, Dr. Demon, for your excellent talk about the perspective and quality control in laboratory 2021 and beyond. So we will move right now to questions. I hope that all the speakers, they are on the floor with us. So just to remind all participants that they have to register for each session, and also they will receive a survey to claim their CME hours at the end of third session. Our first question, actually, Dr. Graham, regarding the in-house developed, as you know, there are in-house developed tests, mainly specialized labs. So regarding the broken chain of metrological traceability and in-house developed tests, what will be the best approach in these cases, and how the accreditation program they are dealing with broken chain of metrological traceability?
Okay, thank you for that question. That is the sound causing a bit of... Okay. Right.
Sorry, Dr. Graham, for the interruption. If the other speakers, they don't mind to put their mic on silence because there are some noise, background noise.
Okay, can we try again? That's better. Right. Okay. It's a good question, and I think for most people that are on this seminar, they won't be able to do anything directly about traceability themselves. What they must do is check the traceability of status of the assays that they actually use. And what you're aiming to do there is to select assays that have as much traceability as possible. In other words, as high up that metrological traceability as it's possible to go. Now, if you're using home developed assays with your own calibrator, then you really have made no progress on traceability, and it's really very difficult in that situation for you to be able to compare your results with those from manufacturers. I understand that if you're using one manufacturer and a platform with lots of assays on that platform, then maybe you don't have a lot of choice about which particular method you use for sodium or calcium or whatever. You really have to use the method from the manufacturer. But I think when you come to change your platform, one of the factors you should consider when you're thinking about a new platform is what is the traceability status of all the assays that can be used on that platform. Now, in terms of broken chains, you're actually right. A high percentage of the things that we measure, particularly things like glycoproteins and so on, it's not possible to get pure material. So you can't get right to the top of the traceability chain. In that situation, what you need to do is make sure that your assay has adopted a conventional calibrator, an internationally recognized calibrator. Now, that might be something from the World Health Organization, or it might be something from another source. But again, if all manufacturers adopt the same international conventional calibrator, although the traceability may not be complete, at least they're all linking against the same particular calibrator. And you would think that would solve a lot of the problems, and to some extent it does. But what we find is that in these broken chain situations, the calibrators that have had values assigned to them are often not commutable. In other words, they don't always behave in the same way as clinical samples. So that when you get into looking at returns on EQA results, for instance, you may find that one particular method is always consistently away from the others when you're using an EQA sample, and maybe not when you're using clinical samples. So it all requires knowledge of what traceability is and an understanding of the assays that you're actually using. But as I said right at the beginning, individual clinical chemists or chemical pathologists can't really do an awful lot about it firsthand. It's really knowing what you're using. I hope that helps.
Thank you, Dr. Graham. Actually, the second question, it can be shared by Dr. Deeman and Dr. Anwar, is related to the QC range. Actually, we have a question if the QC range is broader or is wider than the established provided by the manufacturer. So what will be the suitable action in this case?
Usually, whenever you receive a new lot number of QCs, then you have to validate your QC by establishing a mean and 2 standard deviation before you utilize any new lot number. So at least 20 days before, after establishing your mean and 2 standard deviation, you can utilize your own mean. Usually the companies, they provide you with the mean and 2 standard deviation, but usually they are wider than the one that you should obtain. So you need to establish your own. Later on, if you notice that your QC mean is shifted or your results is shifted compared to the peer groups, then you have to re-establish again your QC and check other factors that might contribute to the shifting in your QC mean. The peer group, of course, it will be very helpful in this case to guide you where to go and what to do. So establishing your mean at the beginning or starting a new lot number is very important in each lab, rather than to use the mean and 2 standard deviation provided by the quality control companies available, or the range available in their kit inserts. This is very important point that need to be addressed to all audience whenever they want to start a new lot number of QC.
Thank you, Dr. Anwar. And again, I think we have another question, Dr. Anwar, related to your advice to use direct method reagent to LDL instead of calculated LDL.
Using the direct methods, of course, it's more accurate and more reliable. But whenever you use the calculated methods within a specific range, as I mentioned in the presentation, that will be also a valid outcomes, valid results that can be utilized. In case, as a general point, I would like to tell you that whenever you can utilize the calculated one, you can calculate the LDL. But whenever the values exceed the limits for triglyceride, for example, more than 400, then the measured one will be more reliable. It depends on your lab and your budget and your so as I said in my presentation, that using calculated methods is one of the procedures that you can follow to save budget, also at the same time to keep reliability and keep patient in a safe side regarding results outcome. So yeah, you can use the calculated one, but within the limitations. If there is if the limits exceed the points, then you have to measure the analytes instead of calculate the test.
Thank you, Dr. Anwar. I would like to remind all the participants to register actually for the next session. And I would like to thank all the speakers, Dr. Graham, Dr. Deeman, Dr. Anwar, for their excellent talk and presentation on one of the important aspects in laboratory practice related to standardization. And that just we will have right now a 15-minute break, and we will get back in our next session, inshallah. So again, to be reminded to register yourself in the next session, and the end of the at the end of our seminar today, you will receive email, a survey that you can claim your CME hours. Thank you all, and we see you in the next session. Thank you.
Thank you.